Our today's topic is ribosomes, polyribosomes, and protein synthesis. Ribosomes are like tiny protein factories inside your cells. They read the instructions from genes, in the form of mRNA, and join amino acids together to build proteins, the molecules that do almost every job in the body: enzymes, structural parts, hormones, etc. Where and how ribosomes are made. Ribosomes are not born fully assembled. Their parts are put together inside the nucleus, specifically in a region called the nucleolus, like a little factory inside the nucleus. There, special RNA molecules called ribosomal RNA combine with many ribosomal proteins, which are made in the cytoplasm and sent to the nucleolus. Once assembled, the ribosome splits into two unequal pieces, subunits. A larger subunit, called 60S in eukaryotic or human cells. A smaller subunit, called 40S. These two pieces travel out to the cytoplasm and float around separately until they are needed for making a protein. When protein synthesis starts, the two subunits come together around the mRNA to form the complete ribosome, about 80S in total. A single ribosome looks like a very small granule, roughly 25 nanometers across, super tiny, millions fit in one cell. In human or eukaryotic cells, the large subunit has 3 RNA strands, 28S, 5.8S, 5S, plus about 49 proteins. The small subunit has 1 RNA strand, 18S, plus about 33 proteins. These numbers are approximate. Textbooks sometimes vary slightly. Free ribosomes versus attached ribosomes. Cells usually have millions of ribosomes because they need to make lots of proteins. Ribosomes can work in two main locations. Free in the cytoplasm, make proteins that stay inside the cell, for example, enzymes for energy, parts of the cytoskeleton. Attached to the rough endoplasmic reticulum, make proteins that will be sent out of the cell or put into membranes or organelles, like secreted hormones, membrane proteins, or proteins for lysosomes, etc. Polyribosomes, or polysomes. One mRNA molecule contains the recipe for one specific protein. Instead of just one ribosome reading it slowly, very often many ribosomes, sometimes 10 to 20 or more, attach to the same mRNA at the same time and all read it together, like multiple workers reading the same blueprint line by line. This chain of ribosomes on one mRNA is called a polyribosome or polysome. The ribosomes move along the mRNA from start to end like beads on a string. Each ribosome makes its own copy of the protein at the same time. Result: many identical protein molecules are produced very quickly from just one mRNA. The more ribosomes are attached, and how closely spaced they are, the longer the mRNA and the bigger the protein being made. What each subunit does during protein synthesis. Small subunit 40S attaches first to the mRNA and helps read the code, finds the start point, and moves along the mRNA. Large subunit 60S joins later and does the actual joining of amino acids, peptide bond formation. It also helps release the finished protein chain. If the protein is destined for secretion or membranes, the large subunit can connect to the rough endoplasmic reticulum via a special docking system, so the growing protein thread goes directly into the endoplasmic reticulum space. siRNA, the off switch, for specific proteins. Sometimes the cell, or scientists, wants to stop production of a particular protein. Small interfering RNA, siRNA, are very short RNA pieces, about 20 to 25 letters long. How they work, simply. The siRNA finds a matching sequence on a specific mRNA, like a key finding the right lock. It teams up with special proteins to form a complex, RISC. This complex either cuts or destroys the mRNA completely, or blocks ribosomes from moving along it. Result: no, or very little, protein is made from that mRNA. In nature, siRNA helps protect cells, for example, against viruses that use RNA. Scientists now use artificial siRNA as medicines to silence bad genes, for example, in some genetic diseases or certain cancers. In short, ribosomes means protein-making machines. Made of RNA plus proteins, in 2 subunits, 40S plus 60S. Polyribosomes means many ribosomes working together on one mRNA, fast mass production. siRNA, tiny RNA. Shutdown. Tools that destroy or block specific mRNAs to stop unwanted proteins. This is the central dogma in action: DNA, mRNA, protein, with ribosomes as the key translators. I hope you liked the video. Please subscribe this channel and press the bell icon.